Okay, let's unpack this. Imagine, um, imagine you are standing at the top of a skyscraper, right? You're looking down at this incredibly vast, super complex map of a bustling metropolis.
Right.
Now, classical epidemiology, which if you're working in public health or clinical medicine, you're obviously very familiar with, it spends all its time mapping the surface of that city.
Yeah, the visible stuff.
Exactly. It maps the roads, the intersections, the traffic patterns. It looks at how close people live to industrial zones or tracks the pollution clouds. Basically, it tracks environmental exposures and how those exposures associate with health outcomes across the population living in the city. But today, we are adding an entirely new invisible layer to that map. We're looking deep beneath the concrete. Genetic epidemiology maps the underlying geological fault lines, the tectonic plates of our biology and our inherited DNA.
Which is such a great way to visualize it, because if we connect this to the bigger picture, that is precisely the mission of our deep dive today. We are bridging that microscopic world of molecular biology with the macroscopic world of population science.
Superimposing those two maps.
Exactly. The environmental traffic above and the genetic fault lines below, because only by putting them together can we truly understand why, a metaphorical earthquake destroys one building but leaves the seemingly identical building right next door completely standing.
Right.
So for those of you listening, whether you're a medical student prepping for boards, a junior doctor on rotation, a public health trainee, or a practicing clinician, you are going to want to have Chapter 5.12 of the Oxford Textbook of Global Public Health open right alongside you.
It's your companion guide for this.
Yeah, this deep dive is designed as an active companion to that chapter. We're going to clarify the conceptual frameworks of genetic epidemiology, unpack the honestly highly sophisticated methodologies used to hunt down these inherited risk factors, and explore exactly how this knowledge is completely revolutionizing public health.
It really requires a massive shift in how we think about risk, doesn't it? I mean, in classical epidemiology, you're generally asking a very straightforward environmentally based question.
Right, like, does smoking cause lung cancer?
Yeah, exactly. Is a measured environmental exposure, like a lifetime of smoking or occupational exposure to asbestos, consistently associated with the specific disease outcome in a population? Genetic epidemiology borrows that exact same rigorous analytical paradigm, but it just swaps out the nature of the exposure.
It looks inward instead of outward.
Right. Instead of asking about the external environment, it asks whether a specific genetic exposure, a variation in your inherited DNA sequence, is associated with a health outcome. But, and this is key, the goal isn't just to find a simple statistical association and just walk away.
No, not at all.
The ultimate goal is to unpick the entire causal pathway. We want to start at the gene, trace it down to the actual biological mechanism, and ultimately provide a priori insights into the disease biology itself, long before the clinical symptoms ever even manifest.
And the pace of change in our ability to do that just over the last decade has been nothing short of staggering. We've transitioned from this era where researchers were painstakingly studying single gene variants in very small localized family samples.
Which took forever.
Took absolutely forever. And now, we are in an era defined by high-throughput genotyping, genome-wide association studies, and next-generation whole genome sequencing. We are routinely processing and analyzing information on millions of genetic variants across thousands, sometimes hundreds of thousands, of individuals simultaneously.
It's massive data.
It is, and it's vital to recognize that this isn't just some niche laboratory science anymore. Genomic medicine is rapidly becoming an essential, everyday tool for public health practitioners.
Yeah, it's not science fiction.
No, it's the core engine driving targeted therapeutics, nuanced risk stratification, and really the future of how we'll address global healthcare needs. If you want to understand how clinical medicine will classify and treat disease in 10 years, you have to master the frameworks of genetic epidemiology today.
Absolutely. So let's start building that conceptual foundation. We have to touch on the biological blueprint itself, DNA, RNA, and proteins. Now, we are obviously skipping the middle school biology recap here.
Right. We all know the double helix.
Yeah, we know the double helix, the ACGT, the basic translation pathway, but let's focus on the differentiation between what actually codes for our biology and what regulates it. Because for a long time, the central dogma of molecular biology was pretty simple. DNA makes RNA, and RNA makes protein.
Right.
But only about 1 to 2% of our RNA transcripts actually encode for proteins. For decades, the remaining 98% was just casually dismissed by a lot of people as junk DNA. So what is actually happening in that vast non-coding space?
Well, that so-called junk is actually the control panel for the entire system.
The control panel.
Exactly. The vast majority of that non-coding RNA has absolutely crucial regulatory functions. It modulates how, when, and to what degree our genes are expressed. And this differentiation is critical for anyone entering this field because it completely changes where we look for disease-causing variations.
Oh, this is not just in the coding parts.
Right. A gene is not just a continuous, uninterrupted strip of protein coding instructions. It's highly fragmented. It consists of coding regions called exons, which are physically interrupted by non-coding regions called introns.
Right.
And we now know, thanks to these massive international genomic consortiums, that variations in these non-coding intronic regions, or even the vast intergenic regions that sit between genes, they can profoundly alter disease susceptibility.
Just by changing the environment around the gene.
Exactly. By changing the regulatory environment, they can alter the timing of when a protein is produced or the specific tissue where it's active, even if the final protein itself is structurally perfectly normal.
And that brings us to how we generate genetic diversity in the first place, right? Which creates the variations we're actually trying to study. You can have genetic variation at 3 distinct levels within an individual.
Let's break those down.
First, you have genome variation. This is the macro level. It affects the actual number of intact chromosomes in the cell, the classic example being trisomy 21 in Down syndrome.
Right, whole extra chromosome.
Exactly. Second, you have chromosome variation. This alters the physical structure of individual chromosomes. This results in things like translocations, where pieces of chromosomes actually swap places, or you get massive deletions. And then third, you have genetic variation. This is the micro level, and honestly, it's what we are mostly focusing on today. These are the changes that alter the specific sequence of nucleotide bases in a defined length of DNA.
And within that third category of genetic variation, we need to establish a very clear taxonomy of variants. Because in genetic epidemiology, the precise terminology you use matters immensely.
It really does.
We're looking at alleles, which are simply the alternative versions of a DNA sequence at a specific physical location or locus on a chromosome. Now, if a particular allele is rare, meaning it's observed in less than 1% of the general population, we strictly call it a mutation or a rare variant.
Less than 1% is a mutation.
Yes. However, if that allele is established and found in more than 1% of the population, we classify it as a genetic polymorphism, or a common variant. The word polymorphism literally translates to many forms, and it just reflects normal, healthy variation within a population.
And the heavyweight champion of these polymorphisms, especially in the context of genetic epi, is the SNP, the single nucleotide polymorphism.
The SNP, yes.
This is just a point mutation where the DNA sequence varies by a single solitary base pair. Maybe you have an adenine at a specific spot while someone else has a thymine. And as of the data noted in our sources around 2012, there were roughly 38 million known SNPs cataloged in the human genome.
It's a staggering number.
It is. But SNPs aren't the only players on the board here. You also have structural variations, things like variable number tandem repeats or VNTRs, where short sequences of DNA are just repeated over and over, and the number of repeats varies between individuals.
Right.
And then you have copy number variants, CNVs. That's where entire substantial chunks of DNA are either deleted entirely or duplicated multiple times. All of these different types of variations work together to create the vast, complex genetic diversity of human populations.
And that diversity gives us our genotype. Your genotype is a specific combination of alleles you inherit at a given locus, one from your mother, one from your father.
Simple enough.
Right. If you inherit the exact same allele from both, you are homozygous at that locus. If you inherit 2 different alleles, you're heterozygous. But having a genotype is just possessing the biological instruction manual. The phenotype is what actually happens when those instructions are carried out.
The physical result.
Exactly. The phenotype is the observable, measurable characteristic or clinical trait. It could be a quantitative, continuous trait like your systolic blood pressure or your fasting glucose level. Or it could be a binary categorical state, like whether or not you've been diagnosed with hypertension. And as any clinician listening knows, the translation from a genetic instruction manual to a clinical phenotype is rarely a simple, predictable one-to-one mapping.
And here's where it gets really interesting. Because the mapping isn't one-to-one, we have to draw a hard, conceptual line between 2 terms that frequently trip people up in the clinic: penetrance and variable expressivity.
Oh, this is a crucial distinction.
Let's use an analogy to keep them straight. Think of penetrance as the light switch on the wall of your living room. Penetrance is essentially the positive predictive value of a gene. It represents the percentage of people who carry a specific disease-associated genotype who actually go on to express the clinical phenotype at all.
Right. Do they flip the switch or not?
Exactly. If a gene has 100% penetrance, every single person with that genetic variant will develop the disease. The switch is firmly wired in the on position. But if a gene has reduced or incomplete penetrance, some individuals with the exact same risk genotype will go their entire lives without ever showing a single sign of the disease. The switch, for whatever biological or environmental reason, just never got flipped.
I really like the light switch analogy. And to build on that, variable expressivity is not the on-off switch on the wall. It's the dimmer slider right next to it.
The dimmer.
Expressivity refers to the clinical range of severity in the features among those who actually do express the phenotype. So let's say you have two different patients who have the exact same disease-causing genotype. For both of them, the penetrance light switch is turned on.
So they both have the disease.
They both have the disease.
No.
But due to variable expressivity, one patient might have incredibly mild, barely noticeable symptoms, a very dim light, while the other patient experiences rapid, life-threatening complications with the light blazing at absolute maximum brightness.
Wow, so same gene, totally different severity.
Exactly. Understanding the distinction between penetrance, whether you get the disease at all, and expressivity, how bad the disease will be, is absolutely fundamental to clinical risk prediction and patient counseling.
Okay, keeping that light switch and dimmer analogy in mind, let's shift to look at the diseases themselves. Historically, the field of medical genetics focused heavily on single-gene or Mendelian disorders.
Right, the classics.
Yeah, these are diseases caused by a mutation in one single gene. They usually feature high penetrance, high clinical severity, and extreme unmistakable phenotypes. We're talking about conditions like cystic fibrosis, Huntington's disease, or sickle cell anemia.
And they track through family trees with very obvious, predictable inheritance patterns.
Exactly. Now, individually, these diseases are relatively rare in the general population, though collectively they do cause a massive amount of suffering. But from an epidemiological standpoint, why did we spend so much time studying them if they're so rare?
Because they're biological windows.
Yes, because they provide incredible, highly focused biological windows. Understanding the exact mechanism of a broken single gene in a rare disorder often shines a massive spotlight on underlying biological pathways that eventually inform our understanding of much more common generalized diseases.
And that is the historical foundation. However, the vast majority of the global burden of disease that public health professionals deal with every day, the cancers, the coronary heart disease, the type 2 diabetes, the essential hypertension, they are not Mendelian.
Not at all.
They are complex, multifactorial diseases. They are polygenic, which means they're shaped by dozens, hundreds, or even thousands of different genes. And crucially, each of those individual genes usually only has a very tiny, modest effect on the overall risk.
Like a tiny nudge.
Right. Furthermore, these genes don't act in a vacuum. They interact dynamically with each other and heavily with environmental and lifestyle factors. In a complex disease, you don't just inherit a single, definitively broken gene that guarantees you will get heart disease. Instead, you inherit a subtle, inherited topographical landscape of risk.
Back to the topography.
Exactly. That genetic landscape might be perfectly stable for decades, and it only triggers an avalanche of clinical disease under very specific, sustained environmental conditions, like a poor diet or chronic stress.
Which begs the methodological question, how do we even begin to measure that subtle genetic influence in something as messy and multifaceted as a complex disease? You obviously can't just trace a clear line through a family tree.
No, it's way too noisy.
Right. We have to look at familial aggregation, but we have to do it with a highly critical epidemiological eye. Yes, families share genes, but families also share diets, they share socioeconomic statuses, they share zip codes, and they share environmental exposures.
Which confounds everything.
Exactly. So, to mathematically tease the biology apart from the environment, genetic epidemiologists use a tool called the recurrence risk ratio. It's denoted in the literature as lambda sub r. This ratio basically estimates the risk of an individual developing a disease, given that their close relative already has it, and compares that specific risk to the general background prevalence of that same disease in the wider population.
And it is a critical metric for establishing familial clustering. But we have to quantify that influence further using the concept of heritability.
Yeah.
represented statistically as H squared.
Heritability.
Yes, and heritability is a very specific, often misunderstood statistical measure. It is the percentage of the overall phenotypic variation of a trait in a given population that is attributable specifically to genotypic differences.
So it's about the variation, not just the trait itself.
Exactly. If a trait in a population has high heritability, it means that genetic variation is the major driver explaining why people in that specific population differ from one another regarding that trait. But to truly separate the nature of heritability from the nurture of a shared family environment, public health has traditionally relied heavily on twin studies.
Ah, the classic twin study design. It's so elegant in its simplicity.
It really is.
We compare monozygotic, or MZ, twins who developed from a single fertilized egg and therefore share 100% of their alleles with dizygotic, or DZ, twins who developed from two separate eggs and share on average 50% of their alleles, making them genetically identical to just standard siblings.
Right.
The underlying logic is pretty straightforward. If you look at twins who are reared together in the same household, you are theoretically holding the childhood environment relatively constant. If, under those constant environmental conditions, you see a significantly higher disease concordance rate, meaning both twins have the disease in the identical MZ twins compared to the fraternal DZ twins, that provides powerful, compelling evidence of a strong genetic etiology for that disease.
The logic is compelling, sure. But as public health professionals, we must constantly interrogate the foundational assumptions of our study designs.
Because twin studies are definitely not without significant flaws.
Not at all. They rely on the equal environments assumption. They assume that MZ twins differ from DZ twins only in the number of genes they share. They assume the environmental exposure within the pairs is practically identical, but does that actually hold up in reality?
Usually not.
MZ twins often share a more identical, competitive intrauterine environment, sometimes sharing a placental blood supply, which DZ twins do not. Furthermore, in postnatal life, society, parents, and peers often treat identical twins much more similarly than fraternal twins. They dress them alike, they put them in the same activities, which subtly synchronizes their environmental and psychological exposures.
So the environment isn't actually equal.
Exactly. Add to this the fact that MZ twins do not actually have perfectly identical gene expression due to somatic rearrangements and epigenetic divergence over time. Finally, there is a massive issue of ascertainment bias. Twins who are very similar in their health and behaviors are historically much more likely to volunteer for longitudinal twin studies than twins who are highly discordant, which naturally skews the data toward heritability.
And those biases make adoption studies another incredibly valuable tool in the epidemiological arsenal, despite them being just logistically brutal to actually pull off.
Oh, they are a nightmare to run.
But the design is fascinating. You identify a specific disease or trait in a population of adoptees. Then you track down and study both their adoptive family, which represents shared environmental factors with zero shared genetics, and their biological family, which represents shared genetic factors with zero shared postnatal environment.
It's a beautiful separation of variables.
It's arguably the cleanest epidemiological way to tease apart nature and nurture. But getting large enough sample sizes for statistical power, combined with the immense ethical hurdles and privacy concerns of contacting biological families who may not wish to be contacted, it just makes adoption studies incredibly challenging to execute on a large scale.
Which brings us to a crucial intersection in our understanding. What's fascinating here is that genetic epidemiology does not aim to replace classical environmental epidemiology.
It's not either or.
Right. It isn't trying to say that the environment doesn't matter. In fact, it seeks to refine our understanding of the environment. The reality is that nearly all complex human diseases originate from a dynamic interplay between inherited genetic susceptibility and modifiable environmental exposures.
Gene-environment interaction.
This is the realm of gene-environment interactions. Understanding these interactions is the holy grail because it answers the fundamental public health question of why two individuals can have the exact same environmental exposure. Let's say they both work in the same factory and smoke a pack of cigarettes a day for 20 years, yet one develops aggressive lung cancer while the other lives to be 90 with healthy lungs.
The inherited genetic factors dictate your baseline susceptibility to that specific environmental insult. But methodologically, trying to study these gene-environment interactions at a population level is an absolute nightmare.
It really is.
To detect a statistically relevant, mathematically sound interaction between a specific gene and a specific environment, the sample size required is exponentially larger than the sample size required to evaluate the main effect of either the gene or the environment alone.
You need massive cohorts.
And we have a massive technological disparity here, too. While our ability to sequence genomes has become incredibly cheap, automated, and hyper-accurate, our ability to accurately and objectively measure complex environmental exposures across massive epidemiological cohorts is still surprisingly primitive.
Oh, the exposure measurement is the weakest link.
Trying to precisely quantify a person's lifetime of dietary intake, their exact exposure to daily microstressors, or their precise physical activity levels over decades relies mostly on self-reported questionnaires, which are notoriously unreliable compared to a DNA sequencer.
That glaring disparity in measurement precision is a major bottleneck for the field. But let's shift our focus to the methodologies themselves. How do genetic epidemiologists actually hunt down and find these disease-causing genes in the first place?
Right. What are the actual tools?
Historically, the first major successful strategy was linkage studies. Think of this as the family tree approach. Linkage analysis relies heavily on the biological reality of genetic recombination. During meiosis, when gametes are formed, chromosomes cross over and swap segments of DNA.
Shuffling the deck.
Exactly. Genetic linkage is the statistical tendency of genes that are physically located very close to each other on a chromosome to be transmitted together to the offspring. Because they're sitting right next to each other, they're simply less likely to be separated by a random recombination event than genes that are far apart.
So, in a linkage study, the researchers are looking for co-segregation. You take a massive multi-generational family pedigree where a disease is highly prevalent. You track the disease through the generations, and simultaneously you track thousands of genetic markers. You're basically looking for a specific genetic marker that always seems to travel with the disease.
Right.
If you find one, you don't necessarily know what the disease gene actually does, but you know the disease-causing gene must be physically sitting right next to that marker on the chromosome. The beauty of the linkage approach is that it requires absolutely no a priori hypothesis about the disease biology.
You don't have to guess.
Exactly. You don't need to know if the disease is metabolic or autoimmune. You are just mapping the risk by physical position on the chromosome.
It was a revolutionary technique, but linkage analysis has severe fundamental limitations. It is fantastic for finding the rare high-penetrance mutations responsible for Mendelian disorders within specific, heavily affected families. It helped us find the genes for cystic fibrosis and Huntington's.
Huge wins for the field.
Absolutely. But it is frankly terrible for common, complex, multifactorial diseases. For a complex disease where dozens of genes contribute tiny amounts of risk, linkage analysis lacks the resolution. It only identifies massive chromosomal regions that might contain hundreds of plausible candidate genes without telling you which one is the actual culprit.
It's like finding the right neighborhood, but not the house.
Perfect analogy. To find the specific causal variants in a complex polygenic landscape, the field had to completely abandon studying families. We had to move to studying entire populations of unrelated individuals. We had to move to association studies.
Let's break down association studies. Instead of looking at family trees, you look across totally unrelated individuals in a population to see if a specific allele is found statistically more often in people with the disease than in healthy controls.
Right.
The earliest iteration of this approach was the candidate gene association study. This operates very much like a classical epidemiological risk factor hunt. A researcher looks at the existing literature and says, based on what we know about the biology of this disease, I strongly suspect that this specific gene might be involved.
You're making an educated guess.
Right. You pick your candidate gene a priori, you find the known variants within it, and you test those specific variants in your group of cases, in your group of controls. If your biological guess is correct, it is a highly efficient and cost-effective method. But the fatal flaw is that it is entirely limited by current human knowledge. You will never, ever discover a novel, unexpected biological pathway because you are only looking under the lamppost where the light is already shining.
The frustration with candidate gene studies is exactly why the field experienced a massive paradigm shift with the advent of genome-wide association studies, universally known as GEDO.
The famous G-way?
GEDO-EOS allows for completely hypothesis-free testing across the entire genome simultaneously. You don't need to guess which gene is responsible, and you don't need to understand the biology beforehand. You take thousands of completely unrelated people who have a specific disease and thousands of matched controls who do not.
Just a massive cross-section.
Then, using microarray technology, you scan their genomes using hundreds of thousands of common SNP markers. You are basically running a massive statistical dragnet, looking for any SNP anywhere in the genome that shows up statistically more often in the disease group.
But wait a minute, earlier we established that there are over 38 million known SNPs in the human genome. It would be economically and computationally impossible to test every single one of those 38 million SNPs in tens of thousands of people.
Yeah.
How are they scanning the whole genome efficiently?
They use a brilliant biological shortcut, and it relies on a concept called linkage disequilibrium. Imagine a massive freight train with thousands of connected cars rolling down a track. If you want to know if the train is currently passing by a station, you don't need to place a sensor on every single one of those thousands of cars.
Because they're attached.
Right. Because the cars are physically linked together, if your sensor detects the engine passing by, you can confidently infer that the rest of the train is attached and passing by as well. Human DNA operates somewhat similarly. It tends to travel through generations in relatively conserved blocks.
So the SNPs travel together.
Many SNPs are strongly correlated with their immediate physical neighbors. So if we map out the pattern of these blocks, we only need to test one representative SNP, what we call a tag SNP, to effectively test the entire block of genetic variation. This tagging strategy makes G2As incredibly cost-effective because you can capture the vast majority of common human genetic variation by testing a fraction of the total SNPs.
And the historical anchor point for proving that this hypothesis-free tag SNP approach actually worked on a grand scale was the 2007 Wellcome Trust Case Control Consortium, or WTCCC.
A landmark study.
It was an absolutely unparalleled undertaking at the time. They analyzed 10 billion pieces of genetic data across 17,000 individuals to hunt for novel genes for 7 major diseases, including coronary heart disease, Crohn's disease, and type 2 diabetes.
And the results were stunning.
It was a spectacular success. They found strong, replicable associations in areas of the genome that no biologist had ever suspected were involved in these diseases. It blew the doors off our conventional biological understanding and opened up entirely new fields of research.
It was a watershed moment for the field. But, as is so often the case with massive scientific breakthroughs, solving one problem immediately revealed a new, more complex problem, the phenomenon of missing heritability.
Right, the math didn't add up.
GWAs was incredibly successful at finding dozens, and eventually hundreds, of genetic variants associated with complex diseases. But when researchers mathematically added up the risk effects of all those newly discovered variants, they found that they only explained a tiny fraction of the total genetic risk that we knew existed based on decades of twin and family studies.
So the obvious question was, where was the rest of the genetic risk hiding?
Exactly. And it turns out GWOs has a massive structural blind spot. It is brilliantly designed to find common variants. But because common variants are old in evolutionary terms, natural selection ensures they only have very tiny effect sizes on disease. GWAS is structurally blind to rare variants, which might have much larger effects on individual risk.
So if GWAS can't see the rare variants because it relies on those pre-selected tags SNPs on a microarray chip, how do we find them? We have to stop using shortcuts. We have to actually read every single letter of the DNA. We have to move from genotyping to sequencing.
Which is a monumental task.
It requires enormous, heavily funded international scientific efforts to build the necessary reference libraries. It started with the original Human Genome Project, which gave us the first baseline. Then the field moved to the HapMap project, which focused on cataloging the common variants to make GWs possible.
And then came the 1000 Genomes Project.
Right. To hunt for the missing heritability, we launched the 1000 Genomes Project, which aimed to deeply sequence diverse populations to catalog much rarer variants, down to about 1% frequency. To truly capture the rare variants responsible for complex disease, the ultimate gold standard is whole genome sequencing on massive epidemiological populations.
The hurdle there is that whole genome sequencing generates incredibly data-heavy files, and historically it has been prohibitively expensive to run on cohorts of 50 or 100,000 people. So researchers developed an elegant, highly efficient alternative: exome sequencing.
Focusing on the coding regions.
Remember earlier in our discussion when we highlighted that only 1 to 2% of the entire human genome actually codes for proteins? That specific 1 to 2% is called the exome. And evolutionary biology tells us that because proteins do the actual work in the cell, mutations in the coding regions are much more likely to cause severe dysfunction.
Because that's where the high-impact stuff is hiding.
It turns out that approximately 85% of all known high-impact disease-causing variants live in that tiny 1 to 2% of the genome. By selectively targeting, capturing, and sequencing only the exome, researchers get an incredibly cost-effective, high-yield way to hunt down the rare functional variants that GWAS inherently misses, while ignoring the 98% of the genome that is harder to interpret.
Now, managing this unprecedented scale of data introduces severe analytical and statistical issues. You mentioned earlier that we don't test every SNP in a GWAS because we rely on tag SNPs. But we can still use mathematical modeling to guess the SNPs we didn't physically test through a computationally heavy process called genotype imputation.
It's an essential analytical tool.
How does that work? Researchers use incredibly dense sequenced reference panels, like the massive databases generated by the 1,000 Genomes Project, to predict or impute the missing genotypes in their own specific study sample. Think of it like assembling a massive jigsaw puzzle. Your GWAS data gives you about 10% of the pieces.
But you have the picture on the box.
Exactly. If you have the completed puzzle box cover from the reference database, you can look at the pieces you do have, compare them to the patterns on the reference cover, and use complex probability algorithms to confidently fill in the gaps. Generating these in silico genotypes drastically boosts the statistical resolution and the power of the study without running new laboratory tests.
And we desperately need that statistical power. In genetic epidemiology, power is everything. Why? Because of the power problem. When you are hunting for genetic variants that only exert a tiny, modest effect on a complex disease, maybe increasing your risk by a mere 1.1 times small sample sizes are statistically fatal.
You just can't see the signal.
Small genetic studies simply do not have the mathematical weight to distinguish a tiny true effect from background noise, leading to massive rates of false negatives. And worse, due to statistical phenomena like the winner's curse, when a small study does manage to find a positive result, it almost always wildly overestimates the true effect size of that gene.
It looks much more important than it actually is.
Yes. To get accurate, reliable data, it is an absolute necessity to have massive sample sizes, often combining multiple international cohorts, and exact, meticulous phenotypic characterization of your cases and controls.
And the need for massive data brings us to the most notorious statistical trap in the field, the multiple testing dilemma. In a classical clinical trial or epidemiological study, if you test a single hypothesis and your GAF returns a p-value of less than 0.05, you generally pop the champagne and consider it statistically significant.
Right, standard threshold.
But in a GWS, you aren't testing one hypothesis. You are simultaneously testing the association of 1 million independent SNPs. By sheer random chance alone, 5% of those tests will yield a false positive. 5% of 1 million is 50,000 SNPs showing a falsely significant p-value of less than 0.05. The statistical noise completely and utterly drowns out the biological signal.
It is a massive problem. To solve it, genetic epidemiologists have to implement incredibly stringent p-value thresholds. You can no longer use 0.05. Instead, they use mathematical adjustments like the Bonferroni correction, which divides the standard p-value by the number of tests performed.
So what's the new threshold?
In GWAS, this usually means requiring a p-value of 5 times 10 to the negative 8th power. That is 0.0000005, just to declare a finding statistically significant. Alternatively, they use computationally intense permutation testing to empirically derive the significance threshold.
That is an incredibly high bar.
But ultimately, you can't rely on statistical gymnastics alone. The absolute gold standard, the non-negotiable requirement in modern genetic epidemiology, is replication. If you find a novel genetic signal in your cohort of patients in London, you must be able to take that exact same genetic marker and replicate the association in an entirely independent, similarly powered cohort in Tokyo or New York or Johannesburg.
Without that, it's meaningless.
Without independent replication, a GWS finding is heavily presumed to be just an interesting statistical artifact.
So what does this all mean? Which brings us to a really crucial pivot. Even if you hit that brutal p-value, and even if you replicate the finding across 3 continents, statistical association is still not biological causation. Just because a SNP is consistently associated with a disease does not mean it actually causes the disease. We have to aggressively interrogate bias and confounding.
We do.
Now, classical epidemiological confounders, things like age, diet, or socioeconomic status, rarely confound genetic studies. Why? Because your DNA sequence is fixed at the moment of conception. It is independently assorted and locked in decades before you ever pick up a smoking habit or get a high-paying job.
That is true. The classical confounders are largely neutralized. But genetics has its own unique, devastating confounder. The ultimate nemesis in genetic epidemiology is population stratification, which is driven by ethnic admixture within your study population.
This one is tricky. Let's break down exactly how this generates false results.
Imagine you are conducting an association study on a population that, unbeknownst to you, is actually made up of 2 distinct, historically separated ancestral subgroups. Let's say subgroup A has a genetically higher background rate of the disease you are studying. Subgroup A also, purely by historical evolutionary coincidence, has a higher frequency of a completely biologically harmless genetic allele. Let's call it allele X.
Okay, so subgroup A has more disease and more allele X.
If you just mix everyone together in your analysis and run a naive association study, allele X will immediately flag as highly associated with the disease. But it's a completely spurious, false positive result. Allele X doesn't cause the disease. It just happens to be a genetic marker for being a member of subgroup A who get the disease more often for entirely different reasons.
It's the ultimate red herring.
Yeah.
So how do researchers solve that and clean the data? They use advanced statistical modeling. One common method is genomic control. Researchers calculate an inflation factor, denoted by the Greek letter lambda. They look at the association statistics of thousands of random genetic markers across the genome markers they know should be associated with the disease. If the population is perfectly mixed, there was no stratification, the average test statistic should be 1. The further that lambda value deviates above 1, the more global bias is present in the population structure.
They use that to adjust the data.
Exactly. They use that lambda value to mathematically penalize and adjust their test statistics downward. Alternatively, a more precise method is principal components analysis, or PCA, using computational tools like Eigenstrat. PCA analyzes the entire genetic dataset to explicitly map and model the subtle, multidimensional ancestry differences between every single case and control in the study. By including these principal components as covariates in the regression analysis, they can completely neutralize the stratification bias.
It is brilliant statistical engineering, but let's take it a step further. Even after you clear away the stratification bias, and even after you replicate the finding, you are still only left with a robust statistical correlation. To definitively prove that a specific gene actually causes a disease, rather than just hanging out in the physical neighborhood of the true causal gene, we need functional validation.
Back to biology.
We have to prove how the variant alters the biology of the cell. This is where massive functional genomics projects like ENCODE come into play. They aim to map the functional regulatory elements of the genome. We need to run experiments to show that the specific variant actually changes gene expression in vitro in a petri dish, or we use in vivo animal models, like creating a knockout mouse missing that specific gene, to see if removing the genetic instructions directly causes the clinical disease phenotype to appear.
And this rigorous demand for proving causality leads us to what many consider the absolute crown jewel of genetic epidemiology, Mendelian randomization.
It really is the crown jewel.
This is the exact point where genetic epidemiology rides to the rescue of classical epidemiology. We know that classical observational studies are perpetually plagued by unmeasured confounding and the dreaded issue of reverse causation. For example, if you conduct a massive observational study and find that people with low levels of circulating vitamin D have significantly higher rates of multiple sclerosis, classical epidemiology struggles to definitively prove the direction of causality.
Did the low vitamin D biologically cause the MS?
Or did having the early stages of MS cause the patients to feel fatigued, stay indoors, and get less sunlight, resulting in low vitamin D as a consequence of the disease?
Mendelian randomization elegantly solves that chicken or egg problem. Here is the conceptual framework, and it relies on Gregor Mendel's fundamental laws of inheritance. At the moment of conception, alleles are randomly assorted and distributed. This means that inheriting a specific genetic variant that naturally and permanently lowers your lifetime vitamin D levels is functionally equivalent to being randomly assigned to a low vitamin D treatment arm in a massive, lifelong, perfectly blinded, randomized controlled trial.
It's nature's own RCT.
Precisely. Because your genetic assignment is locked in at conception, it is completely immune to reverse causation. A disease you develop at age 40 cannot go back in time and change the DNA you were born with. And because genetic assortment is random, it is generally immune to classical environmental confounders.
The textbook provides a perfect, highly illustrative clinical example of this in action: lipoprotein. For decades, classical epidemiology consistently observed a strong association between elevated levels of lipoprotein A in the blood and an increased risk of coronary heart disease.
But the pharmaceutical industry was hesitant to target it.
Right, because observational data couldn't definitively prove causality. Was the elevated lipid level a direct biological cause of the atherosclerosis, or was it merely a harmless bystander, a byproduct of the underlying vascular inflammation? Mendelian randomization finally solved it.
How do they do it?
Researchers looked at tens of thousands of people and grouped them not by their measured blood levels, but purely by whether they possessed genetic variants known to naturally raise lipoprotein A production. They found that the genetically raised lipoprotein A cohort experienced the exact same increased heart attack risk as those with measured high levels.
The genetics predicted the risk perfectly.
Because the genetic variant predates the disease by decades, it definitively proved a causal link, officially validating lipoprotein A as a novel, viable therapeutic target. And to show its dual power, researchers used the exact same Mendelian methodology to refute the long-held belief that HDL cholesterol was a causal protective factor for coronary disease, likely saving the pharmaceutical industry billions of dollars in misdirected drug development.
It is arguably one of the most powerful analytical tools we have developed. But as with all tools, it has biological limitations that must be respected. You have to watch out for a phenomenon called canalization.
Canalization.
This is essentially the body's biological compensation mechanism during early fetal development. If a fetus inherits a genetic variant that lowers a certain protein, the developing biological system might adapt and buffer that defect by upregulating a completely different pathway, masking the true genetic effect.
The body finds a way around it.
The other major limitation is pleiotropy. This occurs when one single genetic variant has multiple, completely distinct biological effects on different bodily systems. If a gene lowers vitamin D but simultaneously independently increases systemic inflammation, you can't be sure which pathway is actually causing the disease, which reintroduces confounding right back through the biological side door.
That makes perfect sense. Biology rarely acts in a perfectly isolated vacuum. But this raises an important question. If classical public health focuses primarily on modifiable risk factors, getting populations to improve their diet, exercise more, and quit smoking, and genes are, by definition, fixed and unmodifiable at birth, why does any of this genetic mapping matter to a boots-on-the-ground public health practitioner or clinician?
That is the crucial transition from theory to practice. The answer lies in how genetic insights completely transform our downstream interventions. The first major, tangible application is in drug discovery and development. When a massive GWAS identifies a genetic variant with a tiny effect size, the clinical utility isn't necessarily in the risk prediction, it is in the biology.
Finding the pathway.
That tiny genetic signal is a giant neon sign pointing researchers toward a massive, previously completely unknown biological pathway. Once that pathway is identified, pharmaceutical companies can develop small molecule drugs or biologics to artificially target and manipulate that pathway, providing massive clinical benefits to the entire population, regardless of their individual genetics.
And it is not just about inventing new drugs from scratch. It is about the massive efficiency of drug repositioning. This is a total game changer for the timeline of clinical trials. Let's look at the specific example of the drug denosumab.
Excellent example.
It was originally developed, extensively tested, and fully licensed to treat osteoporosis by specifically targeting a gene pathway called TNFFF11. Years later, completely independent of the bone research, a GWS looking at the genetics of Crohn's disease found that variations in that exact same TNFFF11 gene were strongly linked to an individual's susceptibility to developing Crohn's.
Linking the drug target to a new disease.
Suddenly, a rigorous biological link was established between the drug's target and the inflammatory bowel disease. This suggested that denosumab could potentially be repositioned to treat Crohn's disease, bypassing years of phase 1 safety trials because the drug was already proven safe in humans.
Beyond discovering the drugs, genetic epidemiology is revolutionizing how we prescribe them through pharmacogenomics or stratified medicine. We have known for a century that patients respond wildly differently to the exact same dose of the exact same drug.
The warfarin example is perfect here.
The classic high-stakes clinical example is warfarin, a very widely used but notoriously difficult to manage anticoagulant. Finding the right dose for a specific patient is walking a tightrope. Give them slightly too much and they suffer massive internal bleeding. Give them slightly too little and they develop a fatal pulmonary embolism.
That's incredibly dangerous.
We now understand that the therapeutic window for warfarin is heavily dictated by genetic variants in two specific genes. CYP2C9, which dictates the speed at which the liver metabolizes and clears the drug, and VKORC1, which encodes the actual target enzyme the drug is trying to inhibit.
Two distinct mechanisms.
Genetic profiling of a patient before prescribing can mathematically predict their exact required dosage, drastically reducing severe clinical complications. Similarly, in infectious disease, genetic polymorphisms at the IL-28B gene locus can predict with genome-wide significance whether a patient with hepatitis C will actually respond to a grueling course of standard interferon therapy, sparing non-responders from months of debilitating side effects.
So we are moving along a continuum of care. We start with risk prediction at the broad population level, then we move to stratified medicine where we group subpopulations to targeted therapies like we just discussed, and ideally we're heading toward personalized medicine with highly customized treatments based on a patient's unique genome.
That's the trajectory, yes.
But we have to be brutally realistic about the current clinical limits of genetic prediction. For common, complex diseases with only modest heritability, genetic profiling currently lacks the discriminatory power to drastically improve a doctor's clinical risk prediction.
It's true, the clinical utility is limited right now.
If you look at the familial relative risk, the FRR, and you map the predictive power of a polygenic risk score on a standard ROC curve, the area under the curve for most GW's findings doesn't beat out classical risk factors. Knowing your advanced polygenic risk score for coronary heart disease is intellectually interesting, but in terms of actionable clinical data, it currently doesn't tell a primary care doctor much more than simply measuring your blood pressure, calculating your BMI, and asking if you smoke cigarettes.
That reality check brings us to the profound ethical, legal, and social implications of generating this data, which public health professionals must navigate. We are generating incredibly sensitive, deeply personal, and permanently identifiable data. At a policy level, we must actively guard against genetic discrimination.
Insurance and employment.
The very real threat of individuals being denied life insurance, health coverage, or employment opportunities based on an inherited statistical susceptibility to a disease they may never even develop. Furthermore, on a societal level, we have to actively combat the fallacy of genetic determinism. This is the false, fatalistic belief that your genetic code dictates an absolute, inevitable outcome, completely ignoring the massive, often overriding role of a healthy environment, preventative behavior, and medical intervention.
Those are massive societal hurdles. And there are equally complex research ethics dilemmas on the ground. How do researchers ensure truly informed consent when the underlying statistical and biological science is this dense and complex? And what happens when you run a massive whole genome sequencing project on thousands of research participants looking for subtle genetic links to type 2 diabetes, and during the scan, you accidentally stumble upon a high-penetrance, highly aggressive mutation for early-onset Alzheimer's disease in a 30-year-old participant?
The incidental findings dilemma.
Do you have an ethical obligation to return that incidental, devastating finding to them? Conversely, do they have a fundamental right not to know their future? And in an era of constant cyber threats, how do we guarantee the protection of this deeply identifiable biological data?
Those questions remain the subject of intense debate. Furthermore, we must address the glaring issue of global equity in genetic epidemiology. Historically, the vast majority of well-funded genomic research and GWAS cohorts have been conducted almost exclusively on populations of European descent.
It's a massive bias.
But from an evolutionary and anthropological standpoint, African populations hold the greatest and oldest human genetic diversity on the planet. If we continue to exclude low- and middle-income countries, or LMICs, from the forefront of genomic research, we are not only missing out on crucial biological insights that require diverse genomes to discover, but we are actively structurally widening global health inequities. The precision medicine and targeted treatments of tomorrow will be perfectly optimized for European genomes, potentially leaving the rest of the global population with suboptimal or even ineffective care.
And it is vital to remember that genetic epidemiology in LMICs isn't just about managing chronic non-communicable diseases. It is an absolute frontline tool for acute infectious disease control. We have to look at pathogen genetics.
Yes, the bugs are evolving too.
For example, targeted genetic association studies recently identified a massive public health crisis: resistance to artemisinin-based combination therapy, the absolute critical frontline treatment for malaria in regions of Cambodia and Thailand. The researchers discovered that this resistance is being driven heavily by rapid evolutionary changes in the malaria parasite's own genome. In under a decade of observation, the heritability of the parasite's clearance rate, how fast it survives the drug, jumped from 30% to an alarming 66%. The pathogen is actively evolving around our best drugs, and genetic epidemiology serves as our global early warning radar.
As we look to the future of the field, genetic epidemiology is doing something even more fundamental. It is actively refining the phenotype. It is redefining our very taxonomy of human disease. Let's take coronary artery disease. For decades, it was treated as a single, broad, clinical entity. But GeoWise is beginning to split CAD into distinct genetic phenotypes driven by entirely different underlying pathological mechanisms, some driven by lipid metabolism, others entirely by vascular inflammation.
It's fracturing the diagnosis.
Conversely, look at autoimmune conditions like Crohn's disease and ulcerative colitis. Clinically, anatomically, and surgically, we have historically treated them as distinct entities. But GWAS has proven a massive, undeniable overlap in shared genetic susceptibility loci, blurring the rigid clinical lines between the two diseases and strongly suggesting a shared underlying pathogenesis that simply manifests differently in different patients.
But at the end of the day, despite all these massive technological and analytical leaps, our biggest hurdle remains risk communication. You would logically think that handing a patient a high-tech, highly accurate genomic risk profile showing they're at severe, elevated risk for developing type 2 diabetes would be the ultimate wake-up call to scare them straight.
You would hope so.
But the human element is incredibly stubborn. Extensive, rigorous studies, including a major comprehensive Cochrane review, have consistently shown that giving people their personal genetic risk data does not reliably motivate them to make sustained, long-term behavioral or lifestyle changes. They might eat a salad for a week, but they quickly revert to baseline. We have successfully cracked the human genome, but we haven't even come close to cracking human psychology.
And that psychological reality highlights a fundamental truth about this field. While genomics is a revolutionary, immensely powerful tool for understanding biology, it does not and cannot replace the fundamental pillars of public health. Genetic epidemiology must be deeply integrated into our broader understanding of the social, environmental, and behavioral determinants of health. It is a tool to help us target our structural interventions where they will be most biologically effective, not a replacement for doing the hard work of improving the environments in which populations live.
Okay, let's unpack one final provocative thought before we transition to our recall prompts. Given how UW-Wells is actively redefining our disease taxonomy, proving that seemingly distinct diseases like Crohn's and UC share deep genetic roots, while seemingly uniform diseases like coronary artery disease are genetically fractured into different mechanisms, will we eventually stop using our classical clinical names entirely?
It's a fascinating question.
Will a patient walking into a clinic in the year 2050 be diagnosed not with the broad term diabetes or rheumatoid arthritis, but instead be diagnosed with a specific pathway for inflammatory defect, a defect that is treated by a precisely matched biologic drug, regardless of whether that inflammation happens to manifest in their joints, their gut, or their pancreas? It fundamentally challenges how we organize the entire historical practice of medicine.
That is precisely the trajectory the evidence suggests we are on. Now, to ensure the conceptual frameworks we have built today are firmly cemented, we are going to transition directly to the single best answer recall prompts. Listeners, use these to test your active recall of the material. I will read the question, pause briefly to let you lock in your answer, and then provide the correct response.
Let's do it. Prompt 1. Question. Which fundamental concept describes the percentage of individuals with a specific disease-associated genotype who actually go on to develop the clinical disease phenotype?
Take a brief second to lock that in.
The answer is penetrance. For consolidation on this concept and how it differs from variable expressivity, reference the heredity, genetics, and disease section of the chapter.
Prompt 2. Question. When utilizing twin studies to evaluate the genetic influence on a complex disease, observing a significantly higher disease concordance rate in monozygotic twins compared to dizygotic twins reared together provides strong epidemiological evidence of what?
I'll give you a moment.
The answer is a strong genetic component or high heritability. To review the assumptions and biases of this method, reference the complex multifactorial diseases section.
Prompt 3. Question. Which specific genetic mapping approach aims to detect an association between an allele and a disease trait across a massive population of totally unrelated individuals, intentionally avoiding the reliance on family pedigrees?
Think about the acronym we discussed.
Answer. Association studies, most notably genome-wide association studies, or GWAS, reference the Finding Disease Gene section for the history of this shift.
Prompt 4. Question. In population-based genetic association studies, what term refers to the confounding presence of multiple ancestral subgroups within a study population with differing allele frequencies, which can cause spurious false positive gene disease associations?
Take a second.
Answer. Population stratification, often driven by ethnic admixture. To review how researchers use genomic control to fix this, reference the causal inference section.
Prompt 5. Question. What elegant epidemiological study design actively utilizes the random assortment of alleles during gamete formation as an instrumental variable to definitively assess whether a biomarker has a true causal effect on a disease outcome?
Nature's own clinical trial.
The answer is Mendelian randomization. Reference the causal inference section to review the lipoprotein A clinical example.
Final prompt number 6. Question. Variation in which two specific genes strongly and predictably influences a patient's required therapeutic dosage of the widely used anticoagulant drug warfarin.
Lock in your answer.
Answer. CYP2C9, which affects metabolism, and VKORC1, which is the drug's target. Reference the Why is Genetic Epidemiology Important for Public Health section.
Here are the details of this episode.
Genetic Epidemiology. Byline. Tracing risk through inheritance where biology meets population science.